So now telophase 1. So during this telophase 1, the chromosomes which have already reached towards two opposite poles of the cell by the end of anaphase 1 start undergoing decondensation, and they undergo decondensation into chromatin fibers. So chromatin network will be formed. Okay, so let's see how this happens. So this is the plasma membrane of the cell. This is the plasma membrane of animal cell. Now this is the chromatin which has now the chromosomes have reached towards two opposite cell, undergo decondensation, and which form decondensation into chromatin network. Chromatin network and nucleolus will be reformed, and even nuclear membrane also reforms. Nuclear membrane also reforms, and this is the centrosome having two centrioles, but astral rays and the spindle fibers got disintegrated. Astral rays and spindle fibers got disintegrated. Okay, let's see these steps. So what happens? So chromosomes which have reached towards two opposite poles of the cell undergo decondensation into chromatin. They undergo decondensation into chromatin network. But these chromosomes do not undergo extended state of decondensation. That means these chromosomes do not form chromatin fibers or chromatin network as in case of interphase nucleus, because again this chromatin has to undergo condensation into chromosomes when the daughter cells of meiosis 1 enters into meiosis 2. And hence these chromosomes are undergoing decondensation into chromatin, but they do not reach the stage of interphase nucleus. Okay, next. After this, nucleolus, which has disappeared during prophase 1, will be reformed, and nuclear membrane nucleolus will be reformed, and even nuclear membrane will be reformed, and all other cell organelles also reform. So nucleolus, nuclear membrane, nucleolus, nuclear membrane, Golgi bodies, ER, endoplasmic reticulum, and all other cell organelles which have disappeared during prophase 1 are reforming during telophase 1. They reform during telophase 1. Now, even during telophase 1, what we discussed is astral rays, and even spindle fibers, they disintegrate. Spindle fibers disintegrate. They disintegrate. Okay, so this is about telophase 1. After telophase 1, cell enters into cytokinesis 1. So what happens during cytokinesis 1? Cytoplasmic division occurs. So telophase 1 is the last phase of karyokinesis 1. After karyokinesis 1, cell enters into cytokinesis 1. So meiosis involves meiosis 1 and meiosis 2. Into meiosis, a cell enters into a meiosis parent cell. When a cell enters into meiosis, it completes interphase, that is G1, S and G2 phases. Then it enters into parent cell, enters into meiosis 1. That means it enters into karyokinesis 1. Karyokinesis 1 consists of all these phases: prophase 1, metaphase 1, anaphase 1 and telophase 1. After completion of telophase 1, now cell enters into cytokinesis 1, which is the second phase of meiosis 1. First phase is karyokinesis 1. Right, so now what happens during this cytokinesis 1? So during this cytokinesis, cytoplasmic division occurs. In case of animal cell, furrow will be formed. So furrows are formed. So in case of animal cells, the cytokinesis occurs by furrow method, by the formation of furrows in the cell membrane towards periphery at the equator. In the cell membrane towards periphery at the equator, not here or here, at the equator. So after formation of this furrow deepens, extends into the cytoplasm of the cell, dividing the parent cell into two daughter cells. So this is about the cytokinesis in animal cell. As these furrows are extending from periphery to center, this is called centripetal type of cytokinesis. Whereas in case of plant cells, so in case of animal cells, cytokinesis occurs by the formation of furrow. So it is called as furrow method or centripetal type of cytokinesis. Centripetal type of cytokinesis. This formation of furrow, what happens, how it deepens, this and all I explained in the previous video, that is during the cytokinesis of mitosis. Now you can go through that video once again. So in case of plant cells, this cytokinesis occurs by the formation of cell plate at the center of the cell. So at the center of the cell, let's say this is plant cell. This is plant cell, this is cell wall of plant cell. So at the center of the cell plate will be formed. This cell plate which is formed at the center extends towards periphery, dividing the cell into two daughter cells. As this division, cytoplasmic division is taking place from center to towards periphery, this is called as centrifugal type of cytokinesis. Centrifugal type of cytokinesis. So after cytokinesis 1, that is by the end of meiosis 1, two daughter cells are formed. Now these two daughter cells enter into meiosis 2. They become parent cells of meiosis 2. So by the end of meiosis 1, these two daughter cells which are formed consists of half the number of chromosomes as that of parent cell. So if the parent cell consists of 4 chromosomes in it, which enters into meiosis 1, and these daughter cells which are formed after meiosis 1 consists of 2 chromosomes in each. So the number of chromosomes is reduced to half, hence meiosis 1 is called as reduction division. Right. Now these two daughter cells which are formed after meiosis 1 enter into meiosis 2. Before they enter into meiosis 2, these two daughter cells undergo a phase, preparatory phase, a kind of preparation during which they synthesize RNA, proteins and other proteins which are required for meiosis 2. That preparatory phase is called as interkinesis. Now in the next class, let's start with that interkinesis.